Welcome to the exciting world of thrombin. In this video, I'll be detailing the structural and functional features that make thrombin such an integral part of the blood clotting process. Its existence was first proposed by Alexander Schmidt in the late 19th century. Thrombin mainly functions as a serine protease. This is a protein-cutting enzyme that uses a serine to digest a peptide bond. Thrombin also is a catalyst for many of the reactions that occur in the blood clotting cascade. Unlike more general serine proteases like trypsin, thrombin has a very specific target. When the blood vessel wall is damaged, thrombin acts on fibrinogen. This converts it from a soluble molecule into its insoluble form, fibrin. These fibrin monomers combine to a fibrin polymer, trapping red blood cells and forming a clot. This clot is shown here using an electron micrograph. Thrombin is normally found in the body in its inactive form, prothrombin. Through the action of the prothrombinase complex, a combination of clotting factors 5 and 10, prothrombin is rapidly converted into its active form, thrombin. Here we see a schematic diagram of the broader coagulation cascade, with enough arrows to get any biochemist excited. Here we show the activation of thrombin and its many targets in the pathway. The following animation clearly shows this pathway. Here we see a tear in the blood vessel wall. This causes the release of tissue factor. Tissue factor activates a small amount of thrombin, which in turn activates nearby platelets, which accumulate at the site of damage. Thrombin also activates factor VIII. On the surface of the platelets, clotting factor VIII binds to clotting factor IX to form the tenase complex, shown here. The tenase complex then activates clotting factor X. Clotting factor X binds to clotting factor V to form the prothrombinase complex. This rapidly converts prothrombin into thrombin, causing what is known as the thrombin burst. This burst of thrombin activity creates multiple fibrin polymers, trapping red blood cells and forming a clot. Here is a brief summary of thrombin's targets. First, platelets are activated by small amounts of thrombin released by tissue factor. Then, thrombin activates coagulation factors 5, 8, and 11 to cause the thrombin burst. Then, fibrinogen is converted to fibrin by proteolytic cleavage. Finally, factor 13 is activated to promote fibrin cross-linkages, strengthening the clot. In order to better understand thrombin, we must now look at its structure. Highlighted here is the catalytic triad. This is comprised of a serine, a histidine, and an aspartate. The structure of prothrombin is also important. Here, we see calcium ions bound to a modified glutamate. This anchors prothrombin to the blood vessel wall, limiting its area of activity to only the site of damage. The rest of prothrombin is attached by a flexible linker chain. Here, we see the complete amino acid sequence of prothrombin. Below, the catalytic domain is highlighted, set in a deep groove, and above, we see the cleavage sites that convert prothrombin into thrombin. This is highlighted in the following structure. As you can see, cleavage by the prothrombinase complex causes an allosteric change in prothrombin. This exposes the catalytic domain and the reactive oxygen of serine. Thrombin is regulated in several ways. Firstly, it has a short half-life. The active form of thrombin only lasts a few seconds. This limits its activity to only the area of damage and prevents clotting from spreading throughout the body. Secondly, thrombin negatively regulates itself by binding to thrombomodulin. This generates protein C, which inactivates clotting factors 5 and 8. This is shown in the broader pathway here. Finally, there's direct inhibition by antithrombin 3, heparin cofactor 2, as well as other non-specific protease inhibitors in the blood plasma. These inhibitors work by blocking the catalytic domain of thrombin. This inhibition is well demonstrated by a different inhibitor of thrombin produced in the mouth of leeches called hirudin. Here, we can see it neatly fits into thrombin like a puzzle piece, perfectly blocking the catalytic domain and preventing it from undergoing its enzymatic activity. I hope you enjoyed this brief look into the structure and function of this fascinating protein. And remember, next time you're bleeding and you're happy it stopped, thank thrombin.